Good morning, boys and girls. Today we're going to be looking at what is going on with antigen recognition by B cell and T cells via their receptors. Looking like this. Why? Because our brains can't handle some of the advanced topography, the advanced folds that may occur, tertiary, quaternary structure of some protein. So we draw it looking like this. It's relatively easy for us to visualize, easy for us to draw. Constant domains. You see here the VH, the CH, CL, VL, variable light, constant heavy, variable heavy, all antibodies, irregardless of which animal they are isolated from, still has those same variable domains and constant domains. Now, when I say domain, what I am talking about is a generalized tertiary quaternary shape within the overall protein. We distinguish domains by using these rectangular shapes here, or the different color codings you see here in the ribbon model. Tertiary quaternary shape within this constant heavy region here means that it's going to be distinct from the continued peptide that comes and forms this region there. They are separated by a peptide strand, a string within the same polypeptide that gives us this domain, that domain. Draw them with this Y configuration. Again, because it's easy for us to draw, it's easy for us to visualize. But we have to keep in mind that that hinge region is not set at the 60-degree angle we like to draw it at. It's actually can expand out further. Here they're showing it going out to 90 degrees and supposedly can even go further than that. It's variable in its angle and it has quite a few degrees of freedom that it can bend through. It is not a set shape. By being able to move, by being able to stretch through different regions, you can see here where it is showing different antigens can be bound up by multiple antibodies, forming this triangle, this square shaped. You get more and more of these binding, you can get larger and larger antibody complexes. Now, as I mentioned before, the structure of the immunoglobulin constant Va and variable regions is set by the secondary and the tertiary as well as the quaternary structure of a polypeptide. Here they're showing you the different subdomains within the variable and this portion of the constant of the light chain. You see the yellow, the green, the blue, the red correspond to different pleated sheets. Pleated sheets that have the turn, sheet, turn, back and forth, as we see here in the secondary as well as the tertiary, and then through the whole thing a relative quaternary structure. This is where we get this idea of immunoglobulin-like domains. Many proteins out there, as we've discussed in previous lectures, previous chapters, are going to have this secondary, tertiary, up to almost the quaternary arrangement of beta strands. By having this back and forth of the beta strands with the turns, giving it an overall three-dimensional shape, the different receptors will have what we will refer to as an immunoglobulin-like or Ig-like domain. And it's all based on these beta strands. Notice here there are very few, if any, alpha helices. You see the beginning of one here, a couple of the turns almost become, but this is mostly going to be based on beta strands or beta sheets. As we're going to see over the next few lectures, it's right here, or the red and the blue, the amino terminus ends of these proteins are going to have the variable region that will have a three-dimensional shape that will be able to interact with not only the receptor, but whatever it was, not only with the B cell T cell receptor, but also whatever's being held as an antigen to trigger inflammation.